In our recent publication, we are trying to better understand how NK cells, cells of the human immune system, are able to recognize hepatitis C virus-infected host cells. And we know from previous work that in this interaction, key HLA receptors play a pivotal role, so we are focusing on that. For this, we use the model system. We use 2-to-1 cells, which are devoid of any human HLA on the surface. They are like a lymphoma cell line, and we transduce those cells with like a specific human HLA molecule. In our case, it was HLA-C or 3 or 4. Those cells then were further modified with a tap block peptide from another virus, which makes it impossible for those cells to internally load peptides on their HLA. This leads to only transient expression of HLA molecules on the surface because there's no peptide which actually stabilizes them on the surface. What we can now do is load peptides of interest, in our case peptides from hepatitis C virus genotype 1, externally on those HLA molecules, and if they are able to stabilize the HLA, this leads to an increase of the stabilized HLA on the surface of the cells, which we then can measure in a number of certain assays. If we then use those peptide-loaded target cells and put them together with human NK cells, we are able to see that if the NK cell expresses the receptor that is able to interact with the peptide HLA complex, their function is inhibited, but if they don't express the receptor, their function is not inhibited, showing us that viral peptides are able to inhibit NK cell function. Or more specifically, viral peptides from HCV genotype 1 are able to inhibit NK cell function. The question now was, how does that look in different genotypes of HCV infection? We found out that in different genotypes of HCV, there are different variants of the sequence that we identified in our first approach, which lead to a sequence-specific HLA stabilization, meaning that the sequence actually determines how much HLA peptide complex comes to the surface or is stable on the surface, which then has further consequences because the more HLA peptide on the surface is, the higher the chances for a strong interaction with key HLA if the cells actually have the key receptor that is able to bind to this complex, showing then that the sequence also determines the inhibition of the NK cell. What this actually means is that there's a highly sequence-specific interaction between key molecules on NK cells and HLA peptide complex on the surface of infected cells, showing that the virus might be able to evade from immune pressure by selecting peptides that lead to a strong binding of inhibitory key receptors to peptide HLA complexes and thereby inhibiting the function of NK cells, preventing the NK cells from killing the virus-infected cells.